Hello, everybody. I'm Megan Swanson, an assistant professor of psychology and faculty affiliate of the Center for Children and Family. It's my pleasure to welcome you all on behalf of Margaret Owen and the Center for Children and Family to our keynote talk for the 14th Annual Center for Children and Family Spring Lecture Series. Before I introduce the speaker, I want to go over a few pieces of business. We are obviously meeting virtually, so I will ask that you please hold your questions until the end of the talk, unless a quick clarification question comes up. Rachel Berglund and I will be monitoring the chat from the team's event, and we'll pass along any questions that come up during the talk to Dr. Noble. People are muted upon entry, and mics will be enabled for the Q&A. So when we get to that session, you can feel free to unmute your mic and raise your virtual hand or put a question that you have in the chat. Our talk today is titled Socioeconomic Inequity and Child Brain Development by Dr. Kim Nobel. Dr. Noble is a professor of neuroscience and education at Teachers College of Columbia University. She's a neuroscientist and board-certified pediatrician, and she directs the Neurocognition, Early Experience, and Development Lab, the NEED Lab, where she and her team study how socioeconomic inequity relates to child's cognitive and brain development. Dr. Noble is a fellow of the Association for Psychological Sciences and was awarded the 2017 APS Janet Taylor Spence Award for transformative early career contributions. Her current research is funded by the NIH, the Valhalla Charitable Foundation, the White Foundation, the Chan Zuckerberg Initiative, amongst many others. She's published extensively on the topic of poverty in the developing brain in journals such as Nature Neuroscience, Pediatrics, JAMA Pediatrics, PNAS, and Biological Psychiatry. If you haven't watched her TED Talk, I recommend you give it a watch. It has received over 2.3 million views and is honestly one of my favorite videos to show in my undergraduate and graduate courses. So without further ado, I'm going to turn the floor over to Dr. Noble. So please, everyone, join me in welcoming her.
Thank you so much, Megan, for that warm introduction. It's really my pleasure to be here today. Good morning to everybody. So the human brain has been termed the most complex 3 pounds in the universe, and it's not hard to see why when we consider the fact that we're born with 100 billion neurons or brain cells, and that we develop between 250,000 and 500,000 new neurons every minute in the first few months of life. But it turns out that early brain growth is not primarily driven by the development of new brain cells, but by connections between those cells. And those brain connections become increasingly complex in the first several years of life. So here you see a picture showing brain tissue under the microscope at birth, at 3 months, and at 2 years of age. And you can really see the remarkable development of the connections between cells. And in fact, we have 1,000 trillion of those connections by the time we're 3 years old. Now, what I find particularly fascinating is the fact that early experience shapes that brain developmental process. And it turns out that the brain is highly plastic or sensitive to experience, especially early in childhood. Now, of course, a child's experience varies tremendously as a function of his or her family's social and economic circumstances. And so we can use social and economic factors as a lens through which to better understand early brain development. So taking a step back, as I mentioned, I study how poverty relates to children's brain development. So what do we mean when we talk about poverty? So poverty is defined as a lack of economic resources needed to attain a minimal standard of living. In the US, the poverty threshold is defined by the federal government according to family size and composition, meaning the number of adults and children in the home. And here, currently a family with 2 adults and 2 children has the poverty line set for them at just under $28,000. And that's true no matter where in the country you live. So the standard federal poverty line doesn't vary according to geography, even though, of course, the cost of living varies quite a bit. Although that's quite a low number to imagine raising a family of 4 on, poverty is unfortunately quite common, especially for children. So I highly recommend this COVID dashboard website that has shown poverty rates since the start of the pandemic. You can see that children across the US have been hovering at around 15% living in poverty compared to 11% of all individuals. So children are almost 50% more likely to be living in poverty than the general population. Now, when we talk about socioeconomic status or SES, which I'll use interchangeably with terms like socioeconomic circumstance, socioeconomic disparities, socioeconomic factors, we're talking about more than just poverty. So I just told you that poverty is strictly defined based on family income, whereas SES is defined according to income, but also other factors like parents' educational attainment, occupational prestige, and subjective social status, or where one sees oneself on the social hierarchy. And it turns out that childhood socioeconomic factors are strongly associated with a number of important cognitive developmental outcomes. Things like achievement test scores, grade retention, literacy, IQ, and school graduation rate, to name a few. But of course, knowing that those disparities exist doesn't really tell us anything about what factors might be contributing to that gap in achievement. And so we can likely think of many factors that are serving to contribute to that socioeconomic gap. Things like differences in access to highly nutritious food, differences in access to high quality health care, exposure to environmental toxicants like secondhand smoke or lead, differences in the home learning environment or schooling or stress. And to this laundry list and the many others you all could likely come up with, I would say yes, really each of these has been shown to contribute in part to the link between socioeconomic factors and children's cognitive outcomes. So how do we make sense of that? Well, one way is to recognize that so-called cognitive skill is really too broad of an outcome, by which I mean that traditional achievement measures like IQ or high school graduation rates aren't specific in terms of brain function, right? We don't have a single high school graduation nucleus in our brains. So instead, by taking a neuroscience approach, we can ask which particular cognitive skills and corresponding brain circuitry vary the most along socioeconomic lines. Now, neuroscience, of course, teaches us that different structures and circuits in the brain support different kinds of cognitive skills. And so again, by taking a neuroscience approach, we can ask which core cognitive systems vary the most with family socioeconomic status. And so that was exactly the approach that my collaborators and I took in a series of studies from almost 20 years ago now, in which we recruited several socioeconomically diverse cohorts of families with children ranging in age from kindergarten on up through adolescence. And across these different cohorts, the results were remarkably similar, where we found pretty remarkable socioeconomic disparities in children's language development and things like vocabulary or early phonological skills or early reading, with more modest but still considerable differences in learning and memory and certain aspects of executive functioning or the ability to regulate one's thoughts and emotions. And so this early work led to some additional questions that my lab has tried to pursue. So number one, how do these differences, these socioeconomic differences in cognition and behavior, relate to underlying differences in children's brain structure and function? Number two, just how early are these disparities in brain and behavior detectable? The reasons that if the first studies looked at children of kindergarten age and we were already seeing pretty remarkable differences, these differences must emerge earlier, but when? Number three, which experiences, and especially modifiable experiences, might be accounting for these socioeconomic disparities in children's outcomes? And finally, how can this work inform interventions? And so for the rest of the talk, we'll touch on each of these questions. I'll show you a little bit of data to give you a taste of how we're addressing each of these. So number one, how do these differences relate to differences in children's brain structure? So our lab has looked at this in a number of different papers. I'm going to highlight one from a few years ago, which at the time took advantage of what was then the largest study to recruit a range of children and adolescents across that age spectrum to really look at what is typical in terms of children's neuroanatomy. Now, since then, there have been other studies that have done this in even larger samples, like the ABCD study, which has found pretty similar outcomes. But I'm going to show you these data, which came from the PING study, or Pediatric Imaging, Neurocognition, and Genetics. And so in that study at the time, they had recruited 1,000 children and teens ranging in age from 3 to 20 from many cities across the United States. And initially, the PINK study set out really just to be a study looking at age-related differences in brain structure across childhood and adolescence. But we were able to take advantage of the fact that these children were recruited from very diverse homes around the US. And so we were able to secondarily ask, to what extent do socioeconomic factors like parents' educational attainment or family income relate to the structure of their children's brains? And here we are specifically looking at the surface area of the cerebral cortex, or the thin outer layer of cells on the outer surface of our brain that does most of the heavy lifting. And so just to orient you, for those of you not used to looking at brain images, this is the front of the brain, this is the back, this is the view along the lateral or side surface. Here again, this is the front of the brain, this is the back, and this is the view straight down the middle or medial surface. And here I'm showing you the average of these 1,000+ children and teens. And so we set out to ask, where in the brain might we see associations between family socioeconomic factors like family income and the structure of the cortical surface? And so the answer was, across nearly the entire brain surface did we find this association. So every point you see in color is a point where higher family income was associated with a larger cortical surface area in children. Now, although we see this link across nearly the entire brain surface, there are some regions shown in yellow where this link was particularly pronounced. And those are regions like inferior frontal gyrus and parts of the temporal lobe that are known to be very important for language development, as well as parts of prefrontal cortex like the anterior cingulate cortex that's very important for executive function, or again, this ability to regulate thoughts and feelings. And so the brain data really recapitulated what we had found previously behaviorally with the strongest socioeconomic disparities in brain regions that support the development of language and executive functioning. Now, there are a few points to note about this. So first of all, this link between family income and children's brain structure wasn't linear, by which I mean that the relationship was strongest among the most disadvantaged children. And in many ways, that makes some intuitive sense, right? If you had a family earning $100,000 a year and you were to give them an extra $10,000 a year, that would surely be nice, but probably wouldn't make a dramatic difference in their day-to-day lives. Whereas for a family earning $20,000 a year, if you were to give them an extra $10,000 a year, that would make a pretty remarkable difference in their day-to-day lives. And the second point, this one is key, certainly people who study child development are very familiar with this idea of individual differences. So there was tremendous variability from one child to the next, by which I mean there were plenty of children from higher income homes with smaller brain surfaces, plenty of children from lower income homes with larger brain surfaces. So although on average across these 1,000+ children and teens, we found this link between family income and children's brain structure, in no way could I know an individual child's family income and predict with any accuracy what that particular child's brain might look like. Okay, so moving on to our next question, just how early are these differences detectable? So the images I just showed you were taken using MRI, which if you've ever had an MRI or used MRI in your research, you know it involves lying perfectly still in a dark and noisy tube, which is fine for adults or older children, pretty difficult when we're talking about very young children, infants or toddlers. So when we look into the brains of our youngest research participants, we pull out another tool in the toolbox, namely electroencephalography or EEG, which instead of measuring children's brain structure, actually can tell us something about children's brain function, the activity of the brain. You can see EEG here is measured using this stretchy cap of electrodes, which are these discs. You can think of these electrodes similar to microphones that are listening in to the brain's activity as the different brain cells are talking to each other. Of course, we're not able to read minds. We don't know what a person is thinking, but we're able to measure the speed of those brain signals. So when we get this signal, we use computer software to break it down into frequency bands so we can see how much high frequency or fast-paced brain activity a person's showing at that point compared to how much slower paced or low frequency activity. And we care about the speed of brain activity for a few reasons. So first of all, we know that on average, as kids get older, they tend to have more fast-paced or high-frequency brain activity. So more fast, less slow as kids get older. However, we also know that kids who are at risk for learning and attention problems often tend to exhibit less fast-paced and more slow-paced activity relative to their same age peers. And finally, we also know from past work that in many cases, kids living in socioeconomically disadvantaged environments or facing other forms of early adversity also often show less fast-paced and more slow-paced brain activity. So a few years ago, this was work led by Natalie Brito, we set out to examine just how early in childhood might we see socioeconomic differences in children's brain activity. So Natalie really wanted to push the envelope and conducted EEG in newborn babies who were just a few days old. And she set out to ask, to what extent might we see differences in socioeconomic differences in resting brain activity within the first few days of life? And her answer was really not at all. So she found absolutely no evidence of any association between either parents' educational attainment or family income and brain activity in any frequency band anywhere across the scalp. So that's interesting, but you might say, well, who knows what's going on in newborn brains? Maybe there's just no variability there. Well, that doesn't seem to be the case. There were different amounts of, in this case, this is high frequency or fast-paced EEG activity. There were different amounts of brain activity. They just weren't explained by socioeconomic factors. Or likewise, you might say, okay, well, maybe there's variability, but maybe that's all just statistical noise, you know, a bunch of differences that are meaningless. Well, that doesn't seem to be the case either, because she subsequently brought these same babies back once they were toddlers, around 15 months old. And then she found that the neonatal brain signal did predict cognitive scores at 15 months. So it wasn't that these differences in very early in life brain activity were meaningless. They just weren't accounted for by family socioeconomic factors. However, in a different study where she recruited infants between 6 and 12 months of age, she did begin to see the emergence of socioeconomic disparities, such that kids from more advantaged homes tended to have more high-frequency brain activity already by the end of the first year of life. So while this certainly is not definitive evidence that this emergence of socioeconomic disparities in brain activity is driven by experience, it at least is plausible that some experiences might be accounting for these differences. And so if that's the case, we can ask, which experiences should we be looking at? So I showed you this slide before. In my lab, we've been particularly interested in two of these, namely the home language environment or family stress. And so I'm going to show you a little bit of data linking those particular experiences to differences in children's brain outcomes. So first, let's talk about the home language environment. Many of you might recognize this slide. This came from the work of Hart and Risley, who in the mid-1990s followed a group of families longitudinally for the first several years of their children's lives. Every month, they went into the homes of these families and for a few hours tape recorded everything that the child heard and said. They then brought their tapes back to the lab where some poor research assistant had to transcribe everything. And they found that at each age, the children who were living in the more advantaged homes tended to hear more words than the children living in the less advantaged homes. And so when they extrapolated that out over every waking hour over the first few years of children's lives, the authors calculated that this amounted to a 30 million word difference in terms of the numbers of words heard by children from the more advantaged homes compared to the less advantaged homes. Now, that 30 million number has been hotly contested in recent years. Most estimates place it at quite a bit lower. Nonetheless, a substantial amount of work has suggested that not only are there differences in the quantity of words heard, but also the complexity and responsiveness of verbal interactions frequently as a function of family socioeconomic backgrounds. And many studies have shown that the number of words children hear tend to be related to their vocabulary and other aspects of language development. Moreover, I showed you a few moments ago that some of the regions where we see the most dramatic socioeconomic differences in children's brain structure are regions that support language development. And so we reasoned that perhaps these differences in the home language environment might account for socioeconomic differences in brain structure in language-related regions. Now, we had it a little bit easier than Hart and Misley and their research assistants were able to take advantage of this technology, which is known as LENA, or Language Environment Analysis. Essentially, it's a small digital recorder that fits inside specialized clothing that the child can wear for up to 16 hours. It automatically records the whole language environment during that time. And then, assuming the family gives the recorder back to us, we can upload the data, and the LENA software automatically tells us the number of adult words the child hears over the course of the day, the number of vocalizations that they themselves make, and the number of conversational turns or back and forth verbal interactions that the child is part of. And so work led by Emily Mers showed that indeed, kids who experience more conversational turns tend to have a larger surface area in this, what we call parasylvian region, this language supporting brain region that we previously found socioeconomic disparities in. And indeed, the difference in brain structure accounted for socioeconomic differences in early reading skills in this sample. Now, I also mentioned we're interested in other experiences as well, one of which being the experience of stress. Now, it's clear that in many cases, families facing socioeconomic disadvantage often face higher stress levels. And we know that when parents are distracted or depressed, that family life is likely to be characterized by conflict or emotional withdrawal, rather than the kind of nurturing and supporting relationships that children so desperately need. Many labs have reported that children growing up in economic disadvantage often have altered levels of stress hormone like cortisol. And we know from many decades of work in both animals and humans that there are certain brain regions that are particularly sensitive to stress and stress hormone. Regions like the hippocampus, shown here in green, which is critical for the development of learning and memory, as well as regions shown here in yellow, part of the prefrontal cortex, the anterior cingulate cortex, which again is critical for self-regulation. So both the hippocampus and the prefrontal cortex have been linked with socioeconomic disparities. And so again, we reasoned that perhaps there are particular experiences driving those disparities. In this case, perhaps differences in exposure to chronic stress might account for socioeconomic differences in the structure of these regions. Now, how do we index chronic stress? A number of ways. Of course, we ask parents about their perceptions of stress, their experience with stressors, and we also measure cortisol or stress hormone by taking a small sample of hair. Now, cortisol is more commonly measured in saliva. There are benefits to both techniques. The reason we like to measure cortisol in hair is because cortisol is chronically secreted in hair, and so by taking a one-time sample about the width of a toothpick, we can get an index of the average amount of cortisol that person experienced over the last 3 months. In contrast, salivary cortisol varies over the course of the day. It can vary by day of the week and according to a number of other factors. And so typically you end up having to get multiple samples of salivary cortisol, which can be a bit more challenging methodologically. Okay, so when we look at hair cortisol concentration, we found that children who have higher hair cortisol tend to have a smaller hippocampal volume, and that's specifically driven in the part of the hippocampus that we know is highly sensitive to stress. And furthermore, this difference in hair cortisol statistically mediated the link between socioeconomic factors and hippocampal size. Okay. So I've tried to present a picture so far whereby we think that differences in socioeconomic circumstance, like parents' educational attainment or family income, lead to differences in experience. And I used the examples of the home language environment or family stress. And in turn, that these different experiences have specific impacts on the development of brain structure and function, which help to shape specific cognitive or behavioral outcomes. Now, if this general model is right and experience matters, it begs the question as to whether this work can inform interventions, and if so, what's the right level at which to intervene? Certainly, we can imagine intervening at the level of cognition itself, most commonly through school-based interventions. We can also imagine, of course, intervening at the level of experience, which could be interventions to increase or promote parent-child talk or reduce stress or optimize nutrition or any of a number of other types of experience-based interventions. But for the remainder of this talk, I'm going to talk about what it might look like to focus on the most distal level at the level of socioeconomic disparities themselves. So work by my social science colleagues has suggested that relatively modest differences in family income early in childhood tend to predict better outcomes for those children. So for example, just a $4,000 difference in annual income between the prenatal year and age 2 has been shown to predict higher cognitive scores, increased adult earnings when those children grow up, increased time spent in the labor force, and even some evidence for improved health in adulthood. But most of those studies are correlational, and as we all know, correlation is not causation, and there are plenty of people out there who would say, well, sure, poverty is associated with these outcomes, but poverty isn't causing all of these different outcomes. It's really all the other things associated with poverty, or even perhaps the choices that parents are making for their children. Now, regardless of where you sit on that question ideologically, from a scientific perspective, the best way to address that is to move past correlation to try to understand if differences in income early in life actually cause differences for children's outcomes. And so that's what we are trying to do with the Baby's First Year study. I'm very proud to be one of the 7 principal investigators of this study. It's a multidisciplinary effort across economics, public policy, psychology, and neuroscience. And so what is it? So Baby's First Years is the first randomized controlled trial of poverty reduction in early childhood in the U.S. The premise is actually pretty straightforward. Back in the spring of 2018, we recruited 1,000 mothers living with low income within a day or two of their giving birth. So we recruited them right there in the hospital postpartum wards. And we did this in 4 metro areas around the U.S. Upon enrolling in the study, moms were told that they would have the opportunity to receive a monthly unconditional cash gift that they'd be free to spend however they would like for the first several years of their children's lives. Critically, though, we randomly divided the moms into one of 2 groups. So the moms in the high cash gift group are receiving $333 a month, which amounts to $4,000 a year. The moms in the low cash gift group received $20 a month or $240 a year. And those amounts were chosen very intentionally. So that difference of approximately $4,000 a year, I told you on the last slide, was associated or has been associated with improved outcomes for kids. So on the one hand, it was an amount that we thought was likely to move the needle. On the other hand, it was an amount that we reasoned would be policy relevant. Now, we began planning the study almost a decade ago. We certainly had no idea that Build Back Better might be on the horizon someday, but we did reason that $4,000 a year is similar in size to many social services and benefits that families living with low income often qualify for. So we reasoned that it was both an amount that might have an impact on kids and an amount that policymakers would sit up and take notice about. The money is distributed on a debit card that we activated in the hospital and that automatically gets reloaded each month. The moms get a text message with each reload. And to date, we've put more than $5 million into the pockets of the mothers in our study. And so by comparing the differences in outcomes in the children of the moms in the high cash gift group versus the children of the moms in the low cash gift group, we can actually assess the causal impact of poverty reduction on early childhood cognitive, emotional, and brain development during the period of life when we believe that the developing brain is most malleable to experience. Now, how do we think that these increased economic resources are likely to make a difference for the families? We've got two main pathways we're hypothesizing. So the first we call the increased investment pathway, and that's the idea that with greater economic resources, moms are more likely to be able to afford things like books and toys, perhaps higher quality housing in safer neighborhoods, perhaps higher quality childcare. And the other pathway we're calling the reduced stress pathway. And that's the idea that if moms are less worried about how they're going to make that monthly rent check or keep the lights on, they're more likely to have the bandwidth to be able to engage with their children in the kinds of warm and nurturing ways that promote child development. Now, the wonderful thing about money in many ways is that it's fungible, right? So different families can use it for different purposes. So although we're measuring these pathways carefully as we go along, it's quite possible that the exact pathway might vary from one family to the next, depending on what they use the money for. And so ultimately, our primary outcomes of interest really center on the impacts on the children. Okay. So with apologies for this busy slide, this is just a schematic showing where we are with the study. So as I mentioned, we began recruiting the families in the spring of 2018. We recruited them over the space of a year, and we've then followed up with the families annually at around the time of the children's birthday. So for the age 1 data collection, we were initially following up with the families in their homes. So we would request to visit them in their homes to administer a survey to the moms. We did a 10-minute parent-child free play that we video recorded for subsequent coding of parent behavior. We took a measure of mom's hair cortisol, got some maternal report measures of language and social-emotional development of the infant, and some data I'll show you in a moment. We took a measure of brain activity using mobile EEG, so EEG actually in the home. Things were going along great until, of course, March of 2020, when the pandemic struck. Obviously, at that point, we were no longer able to travel into the families' homes. So unfortunately, all of the great home-based in-person data collection, things like videos and hair and, of course, infant EEG, we weren't able to continue to collect. We were able to continue to collect the survey by phone. So the good news is we pivoted quickly. We were able to retain about 93% of the sample overall across age 1, which is great. But our numbers are much smaller for those in-person data points, unfortunately. We had originally hoped to resume home visits at age 2, but that wasn't in the card. So the age 2 data collection was again entirely done by phone. And age 3 was initially planned to be our big capstone wave of data collection, where we were going to invite the families into our university labs to do a thorough in-person direct assessment of cognition and behavior and brain development. Sometime relatively early on in the pandemic, we realized that even at age 3, that might not be feasible. So we went back to our funders, who very generously agreed to extend the cash gift for an additional year through age 4. So that meant that we were able to turn age 3 into a phone survey as well. That's ongoing right now in the field, and postpone that capstone wave of data collection to age 4. And so we've been spending the better part of the last year busily planning for a resumption of in-person data collection, which is set to launch this coming July. So we'll be inviting the families into our university labs to conduct assessments of language and executive function and early literacy skills, as well as brain activity and various other metrics that I'll tell you a bit about later. Okay. I mentioned that the cash is dispersed on a debit card. It's branded the For My Baby card. And the disbursement is automatic, and moms get a text message about reminding them of each disbursement. Now, importantly, I've been using the term cash gift, which is very intentional. It's truly a gift. They can decline to participate in research and still receive the money. That's important from an ethical participant's perspective, in that we can't coerce families to participate in research. It's also important from the perspective of something known as clawback. So clawback is the idea that if I were to give someone $333 a month, that money may render them ineligible for other social services that they were getting, something like food stamps or Medicaid, for example. And obviously that would be an ethical problem, and it would also be a scientific problem if our high cash gift group was systematically losing benefits. So a great deal of time was spent before the launch of the study ensuring that, to the extent possible, moms would not lose eligibility for other social services and benefits. Because it's a cash gift, that means it's not taxable, and there are many benefits tied to taxable income. And for other benefits, this was work led by my colleagues, Catherine Magnuson and Lisa Genetium. We got in 2 of the 4 cities agencies to agree that, for these moms in the study, this won't count in determining their eligibility for social services and benefits. In the other 2 states, we actually got state legislation passed to that effect, that the baby's first year's money won't count in determining their eligibility, so they won't systematically lose benefits as a result of participating. Okay, now I'm sure you're very eager to see some of the early impacts. I am going to talk a bit about our first impact paper, which you may have seen came out a couple months ago. So this was looking at the early impact of 1 year of cash gifts on children's brain activity. And I'll tell you, we've got some other papers in the pipeline from early findings as well, but I'm going to tell you about the published results today. So first, before I show you the results, I have to give a huge plug to the amazing Sonia Troller-Renfrey, who really spearheaded this effort. Truly, it was an unprecedentedly successful mobile EEG data collection effort in families' homes. Usually when we conduct EEG studies, we're doing it in the, comfort and relatively controlled setting of the university lab. Things are quiet, there are minimal distractions. Obviously that's not the case when you're traveling into homes with siblings and pets and whatever else. But we were able to pull it off. So the vast majority of families who we visited in their homes agreed to try it. Out of those in-person visits, 84% of the infants were successfully capped, ultimately yielding high-quality usable data in 72% of the in-person sample. So those numbers are really similar to what we might expect from a lab-based study, so we're really pleased about that. Of course, as I mentioned, the pandemic unfortunately cut us off sooner than hoped. So our final N was only 435 out of the 1,000 families from whom we were able to obtain high quality infant EEG. Now, a little reminder, what were we expecting here? So past work has suggested that children raised in poverty or facing other forms of early adversity often show less high frequency or fast paced activity and more slow paced or low frequency activity relative to their same age peers. And we care about that because that's a pattern that's been associated with subsequent learning and attention problems in school. And so we reasoned, hypothesized, and pre-registered that the cash gifts might reverse that pattern, that by reducing poverty, we would potentially see more fast-paced or high-frequency brain activity and less slow-paced or low-frequency brain activity in the infants of the moms in the high cash gift group. So what do we see? So to walk you through this here, on the x-axis, I'm showing you frequency, so from low frequency to high frequency. Okay. On the y-axis, I'm showing you EEG activity in power, and this is Z-scored. So a score of 0 means they're at the average. And here, the black line represents the infants of the moms in the high cash gift group, and the gray line represents the infants of the moms in the low cash gift group. And so what did we predict? So we predicted at the low end of the frequency spectrum that the high cash gift group would have less low frequency than the low cash gift group. We do technically see that, but it is a very imprecise measure, so not even close to statistically significant here. Then we see this crossover whereby in the mid to high frequency range within every single Hertz bin, the infants of the moms in the high cash gift group are consistently showing more high frequency power, more fast-paced brain activity compared to the infants of the low cash gift group. So compelling descriptively so far. Now, when we actually measure brain activity using EEG, we don't usually measure it as a continuous variable the way I'm showing you now. Instead, canonically, the EEG spectrum is broken down into frequency bands that are very helpfully labeled with literal Greek letters. So this is actually in Greek, where theta represents the lowest frequency, alpha is kind of mid frequency, beta is high frequency, and gamma is very high frequency. And so we can take a look at those individual frequency bands. And so what we predicted was that the high cash gift group would show less theta, more alpha, more beta, more gamma. Okay. So what happens when we take a look by frequency bands? Well, really no difference in theta, more alpha, but not quite statistically significant, and significantly more beta and gamma. So, some modest support of our initial hypotheses. However, when we control for multiple comparisons, the significance of these differences falls. Again, remember that we had about half of the sample that we had hoped to have at age 1. So we did some digging to try to understand for ourselves to what extent are these differences likely spurious versus likely a true signal that we were underpowered to detect. So we did a couple things to try to investigate that. So first of all, instead of looking separately at mid, high, and very high frequency power, we created a composite. When we do that, we do see significantly more mid to high frequency power in the high cash gift group compared to the low cash gift group. And because you're just looking at one outcome, there's nothing to multiply correct for. Moreover, we can take a look at where in the brain these differences are driven from, right? If they're driven by differences that support higher order cognition, that's much more compelling than if they're driven by, lower sensory regions, for example. And so here I'm showing you on the left, the high cash gift group, on the right, the low cash gift group. Beta is high frequency and gamma is very high frequency power. And these circles represent the average of these signals across the scalp, across the group. So if you can imagine someone lying down with their feet into the screen. So this is the back of their head, this is their front, that's the nose, and these are the sides. And so we can map where across the scalp we see high frequency and very high frequency brain activity in the high cash gift group versus the low cash gift group. So first, let's take a look at high frequency or beta activity. You can pretty clearly see that visually we're seeing more of this high frequency activity in the high cash gift group compared to the low cash gift group. And that's specifically being driven by frontal and central regions. Likewise, for the very high frequency gamma band, again, we see differences with more gamma power, more very high frequency power in the high cash gift group compared to the low cash gift group, again, being driven by frontal and central regions. And notably, these regional differences remain significant even when adjusting for multiple comparisons. And so looking across the weight of the evidence, we concluded that this early evidence from the Baby's First Year study really does suggest that reducing poverty may cause differences in children's developing brains in a pattern associated with the subsequent development of higher cognitive skills. So we feel this is really the strongest evidence to date that income may matter in and of itself for brain development, that changing income may change the way children's brains develop. Clearly, we were limited in our statistical power because of the pandemic, so we're very much looking forward to being able to measure this again as kids get older. So what's next for Baby's First Years? We're currently analyzing the age 2 wave of data collection from the phone survey. We're administering the age 3 phone survey. And I think post-pandemic was probably a little overly optimistic, but we are very happy to be launching the age 4 wave of data collection in person this summer, where again, we'll have in-person measures of cognition, social emotional development, brain function, including both resting EEG, which I showed you here, as well as task-based event-related potentials. For those who are familiar, we'll be measuring stress hormone both in moms and kids, measuring body mass index in moms and kids. Again, we'll be capturing parent-child activities. We'll be getting a measure of mom's executive function, and we'll be collecting an epigenetic swab from both moms and kids. Now, we're often asked, what are the policy implications here? And it's our hope that we'll have the potential to provide evidence of the effects of poverty reduction on the developing child and family life across many spheres. It's clear that the policy landscape has changed dramatically. When we first set out to do this 10 years ago, we were told, never would there be the idea of unconditional cash transfers at the federal level. Now, of course, there have been many instances of universal basic income and other child allowance type policies scattered across the U.S. And for a year, the Biden administration expanded the child tax credit so that it was fully refundable, meaning even our poorest citizens who don't owe taxes and therefore wouldn't have benefited from the traditional historical child tax credit were able to benefit from this unconditional cash transfer. And so it's our hope that the results of our study will inform debates on the generosity or cuts both to existing safety net programs as well as new tax refunds and child allowances that are being debated in Congress. Because while we would never suggest that income is the only or even the most important factor in determining child development, it may be one that's particularly easily changed by policy. So with that, thank you very much for your attention. Enormous gratitude to my collaborators, co-investigators, wonderful lab, and of course, our generous funders.